Okay, so in this video, I want to talk about how action potentials get to become neurotransmitter signals. So first off, if we have over here in the dendrites, these dendrites, if they get an EPSP, so they get a signal from another neuron, they're going to generate some current or here, and it's going to depolarize the membranes. If there's enough of a depolarization for it to be felt over here at the axon hillock, then we'll have a signal sent down here to the axon terminal. And remember, if there's not enough, there will be no signal. And that's important in sort of how our neural circuitry computes stuff. But for now, we're just going to assume that the signal gets down here. Now here we have voltage-gated ion channels, which is different from what we have up here. The voltage-gated ones, when they sense a change in voltage, are going to open up, and they're going to create essentially more change in voltage. And that's going to be enough of a change in voltage to get over here to the next non-myelinated space, where other voltage-gated ion channels can create a greater voltage. And this will propagate the voltage change all the way until it gets down here to the axon terminal. And so these nodes of RANVA are the parts between the myelination here. These are the myelination, so the Schwann cells or the oligodendrocytes, whatever sort of neuron we want to say that we have here. So let's say that we get our voltage, the signal, all the way down here. What we'll actually have is we'll have voltage-gated calcium channels at the end. And that's different from up here. Up here, it wasn't so important that it be calcium specifically that gets into the cell. What we really need is we need the cell, the voltage to change. Here it has to be voltage-gated calcium channels because the calcium is actually going to react with some, or sort of bind to some proteins in here and cause vesicles to release neurotransmitters. So it's going to change what was an electrical signal into a chemical signal. And we'll do that in our next video. I want to talk a little bit about how we do that. But for now, you just need to understand that we get all the way down here as an electrical signal. And then once the voltage is sensed here by these voltage-gated calcium channels, then this is where we're going to change it into a chemical signal by letting calcium into the cell.